Hi everyone, I'm Chun-I and I'm from the National Taiwan University. Thanks Brett and John for inviting me to give this lecture, and I'm thrilled to be here because I'm really a novice in this field. So what I'm going to do is I'm presenting what I do in the study. So this study is funded by ZAP medical system, surgical systems, and then, but I personally did not receive compensation from this study. So the topic of my talk is chronic metabolic effect of focal radiation in a Minipig model. So there are three key things in my study. One is focal radiation. So we are going to radiate a very small region of the brain. The second is in a Minipig model because we are using a normal healthy brain. The third one is chronic effect. So we are following up the animal situation for what I say would be 2 years. So yes, it's a longitudinal studies. So I think my results could answer some of the previous questions. Okay, let's see. Okay, yeah. Okay, so previous speakers already mentioned some funding in the focal radiosurgery. So it's been found that radiosurgery is a potential neuromodulation tool that has been used to treat brain disorder besides tumors, including OCD and trigeminal neuralgia and epilepsy. However, the underlying neuronal mechanism is still largely unknown. And also what's the long-term effect of the SRS is also unknown. So the main goal of the current study, the first is to determine the dose-dependent effect of SRS on a small region of the normal brain. The second is we are using the MRI and histology to detect a structural change. Third, we are using the PET signal to detect the change of metabolic activity. So we use the follow-up MRI and PET imaging to reveal a long-term effect of the SRS for more than a year. So we wonder that how long does the SRS-induced metabolic change sustain? And is the SRS-induced metabolic change relevant functionally? That will be using a diction model that. The animal we are using is little mini actual pigs. It actually is raised endogenous in Taiwan. So it's a mix of the Lanyu and the Landrace pigs. So the mini pigs, here you can see the size of the mini pig. The gray on the back is 10 centimeter in length and width. So you can see that this is the pig is about 1 year old, and the size of the pig is about 60 to 70 centimeter when the pig is 1 year old. On the right side is a demonstration compare the brain of size of different species. So on the most left is the human, and for a mini pig and the monkey, they have a very similar brain size. Is 6 centimeter by 7 centimeter. And in contrast, in the rodent, the brain size is relatively small. So the reason that we're using the pig brain is because it's large enough for the application of SRS on a specific brain structure. Consider the position of the SRS technique and the size of a cone colorimeter. Second is the mini pigs is the brain is more similar to humans than rodents. Here I give you an example. For example, the volume of neural cortex in human and the monkeys and mini pig is larger than 70%. In contrast, in rodent, it's only less than 30%. The third one is the pig model is suitable for the long-term research. The mini pig, the Lanyu pig, they mature at 6 months, and then the volume of the brain change is only 10% in the next 12 months and remains stable thereafter. So then I'm going to go through the experimental procedures. So to do the radiosurgery on mini pig, first we need to implant some fiducial marker before the radiosurgery in order to let the multiplane can capture the structure. So here in the figure, you can see a 4 small black dot. That's the place we place the fiducial markers. And then we did a baseline MRI and the PET scan. And then we did the intervention on the primary visual cortex. I will explain why we choose the primary visual cortex later. And then after that, we do the follow-up MRI and PET scan at the days 7, 90, and 180, and 270, and 360. So we use 7 different pigs, and then they receive different doses. Here, like the dose from the 10 to the 100 gram. So that's the Dmax. So actually, the effect dose are within the 80% ideal dose line. So it's 80% of the Dmax. So there are 2 key things here. One is that we choose the 7.5 cone kilometers for select. Because this is actually a suggestion from a PET specialist, because she told us that the resolution of the PET signal is 4 millimeter. So if we use the 5 millimeter cone kilometer, probably it's too small to detect a change in the PET signal. So we choose a relatively larger cone kilometer in this study. So in this figure, you can see that the orange circle represents 7.5 millimeter cone kilometers. The second is that we choose the primary visual cortex because we want to assess the possible behavior deficit of radiosurgical treatment. And in my opinion, the motor cortex or the motion is probably can be easily detected in the pig model. And what we found is that for the animal receive a dose higher than 80 and 50, they seems to show some motor deficit in their hind leg, because this is the region corresponding to the hind leg. So first, I'm going to show you the MRI result. So we did different sequences of MRI image, including T2 weighted and the flare and the T2 weighted. Here I show you an example that the animal received 80 gray in the motor cortex. You can see after 270 days, the deficit show up in the left primary visual cortex, primary motor cortex, but there is no change in the right primary visual cortex. So so next I'm going to show you all of the animals. So first I show you the animal receive a low dose, and on the x-axis is the different doses. On the y-axis is different follow-up days. So what you can see is that basically even as high as the 40 gray, you didn't see visible change in their T2 signal. So here I'm going to show you the T2 weighted image, because the T2 weighted image has the highest contrast. So in contrast, in the higher dose, like the 60 and 80 and the 100, we did see some change at the later day in their left primary motor cortex. So because T2 detects the CSF, it becomes brighter in these regions. So to quantify this change, we did calculate change index. So the change index is a ratio between the left ROI, it means that the irradiated area, and the right ROI is the non-irradiated area. So if there's no change, then the ratio should be close to 1. If there's a change, the larger the ratio means that the change is larger. So I'm going to show you the data at the different date. So here on the y-axis, I represent a normalized change index because we divide the number in the baseline. And on the x-axis, I show you the animal that receive different Dmax. And on the right side, I also show you the negative control is actually in the thalamus. So we also calculate the ratio between the left thalamus and the right thalamus of the 7 animals. And then this is the one standard deviation I'm showing you here. So in this figure, you can see that after 90 days of receiving the 1 dose of focal radiation, the MRI image did not show a significant change. But after 6 months, we did see a significant change at the Dmax equal to 100. So you can see that, but still didn't see that in the 60 and the 80. So and then on the follow-ups 270, we see a significant change in the dose 80. So it seems that the higher dose, we can see the deficit or injury earlier compared to the relatively lower dose. So in the 80, they kind of peak at the 270 days. So one of the reasons for this point to drop is because actually when we go back to check their MRI image, it becomes darker is because actually the place is filled with the blood clot, so it becomes darker. So but it clearly damaged at the 100 gray level. And then on the 360 day, we also showed a significant change at the 60 gray. So based on the T2 weighted image, this is the two main findings that in the T2 in the MRI image. The first is the dose effect. We find the injury at the higher dose. The dose seems to be larger than 60 gray. But for the lower doses, we didn't see much change. You can see that the level of the change is similar to the baseline. So also we see a time effect. Okay, the injury appear earlier in the 100 gray than in the 60 gray. So it seems that the larger the dose, the earlier that the brain is injured, that we can is identifiable injury is found. So this is the result from the MRI techniques. But you may wonder that whether the MRI technique is not sensible enough to pick up the change in the brain under the lower dose of irradiation. So then we did the histology. So the way that we did the histology is that we first we perfuse the animal and then cut the brain to several different chunks. Then the second, we find the best match images of the MRI with this chunk. So in this case, is the 37 here. And actually, the yellow square represents the center of the irradiation. So let me enlarge the image of 37 to show you that we can really see the match. So you can see that in here, the white matter is represented in white, but in contrast, in the T2 image, it actually stopped. So you can see that we can, based on the shape, to find where is this area or this part of the brain. So because we are using the 7.5 cone kilometer, so actually from slides 38 to 44 are the range of irradiation because we are doing the 1 millimeter isolation. So actually it's a very large area, so we can find the tissues. So here I'm going to show you one of the example. And I forgot to mention that we did a histology actually is 18 to 24 months after the radiosurgery. So here is one of the histology example. So this is the picture. You can see that this is the right M1. You can see that based on the white matter is matched pretty well. So this is the slice from this region. And in the center here is the white matter, and then on the two sides is the gray matter. So what you can see is that basically you didn't see any damage in this region, but in the left side, you see a smaller region in some of the part. You probably want to guess, okay, so, and the green represent the stem of the neuron stem. So basically it's a stem, the nucleus of the neuron. So you probably want to take a guess that what would be what's the dosage of this animal. So let me show you the main result here. So here I show you the 4 different animals. They received the dosage from 60 to 10. And I first show you the left hemisphere of irradiation, that the hemisphere that received the irradiation. So what you can see is that for the 60 gray, you can see that the region is, you see a very large injury in M1. And for 40, the injury seems to be spotty. And for 10 and 20, we did not detect any injury in the histology. And on the right side, this is the control M1 that does not receive the radiosurgery. So you can see that even as high as 60 gray, you did not see any change in the brain tissue. So since we are doing the histology in the 18 to 24 months, so it is possible that, for those animals that receive a lower dose, maybe the brain, repair itself, just like the previous speaker said. So that's one of the possibilities. So maybe in the future, we should do more histology at the earlier time to check whether this is agree with our result. So next, I'm going to show you the PET signal. So the PET signal is, we are going to show you the SUV, the standard, is the standard uptake value of the PET. So the glucose we are using is the 18F fluorodegrose. So for the PET, let me talk to you about a little bit about the experimental procedures. For humans, most of people receive the PET scan when they are awake, but it's very hard for the animals because they kind of move around. So we need to keep them kind of in the awake resting condition. So we use the 1% isoflurane. So maybe, the isoflurane can lower, can reduce the metabolic activity, but that's one of the possibilities. And the scan time is about 35 minutes. So here I show you the first 4 animals. They are receiving a low dose of radiosurgery from 10 gray to 40 gray. And then here I show you the data from 1 day after radiosurgery, 90 days, 180 days, 270 days, and 360 days. So what we see here is that we did see some elevations, especially after 90 days of the radiation. And then the elevation seems to last for a long period of time. So in contrast, for those animals who receive a higher dose, we see a decrease in the metabolic activity, especially in the higher dose like 80 gray and the 100 gray, in contrast to the control M1 in the right hemisphere. So again, we are calculating change ratio as the ratio between the left hemisphere to the right hemisphere. And then it means the region between the yellow circle and the green circle, and the size of the circle represents a 7.5 millimeter of kilometer, home kilometer. So again, I'm showing you the data at the different day. First, I'm showing you at 3 months after the radiosurgery. So again, on the right side, I show you the negative control at the thalamus. So the ratio should be equal to 1 if there is no difference between the left hemisphere and the right hemisphere. So what you can see already that there's a small elevation in the lower dose and decrease in activity in the higher dose, even as early as 3 months after radiosurgery. And then this trend is become more clearly after 6 months. You can see that the elevation in the low dose and the decrease in the higher dose. And for the negative control, you see the ratio is still very close to 1. And this is the data from 270 days. You can see that for those animals receive a very, very high dose, the activity is largely reduced, but the activity increase in the lower dose animals remain. And then even after 1 year of 1 dose radiosurgery, we still see some elevations for the low dose population and decrease in the high dose population. So here's a brief summary of the result from the PET signal. So we see a dose-dependent effect, increase at low dose and the decrease at high dose. And also we see a time effect. We find the SRS-induced metabolic change could last for several months. You can see that the effect can happen as early as 3 months. So here is the Intrinsic summary. So we found that focally delivered ionic radiation of low dose, a low Dmax smaller than 40 gray, could induce durable and elevating metabolic activities without leaching the brain tissue in the process. So the PET ratio was significantly larger than the pre-radiation baseline at several follow-up time points. More important, there was no identifiable injury based on the T2 weight MRI image and the histology. In contrast, the irradiation region in the high-dose animals, Dmax larger than 60 gray, shows significant reduction in metabolic activity and detectable injuries. The larger the irradiation dose, the earlier the injury appears, and the higher the severity of the injury is. So, the next question is that is the 10% change of the PET signal functionally significant? I think that's the most interesting is that whether the radiosurgery could be a modulation tool that causes the functional change. From the earlier talk from the Kathy, we already see that yes, by changing the neuroactivity, we can in the nucleus compass, we really can change the people's appetites and also even their mood. So then we established another addiction model in the Minipig is the alcohol addiction model. So here I'm going to show you very preliminary results from our addiction models.
Youngie, before you leave,
Yeah.
Before you leave, can you comment on to what extent is metabolic activity correlated with circuit activity, neuronal circuit activity?
Sean, I cannot hear your question. I'm sorry.
Chun-I, to what extent does metabolic activity correlate with neuronal circuit activity? Can you comment on that?
That's also one thing I'm thinking here. It's very hard to quantify the PET signals. So, for example, what are the 10% metabolic change corresponding to the neural activity change? That's what I really don't have the answer. And also, our animal is actually under the isoflurane anesthesia. So the change could be even higher, and I believe that there's a positive correlation between the metabolic change and the neuronal activity, although we don't know whether the neuronal activity change is in the inhibitory neuron or excitatory neuron. And previous speaker seems to suggest that it could be the inhibitory neuronal circuitry.
Thank you.
Did I answer your question?
Yes.
Are you finished and ready for questions, or did you have more?
So let me give me 5 minutes to show the show clip to show you the animal the Minipig the alcohol addiction model in the Minipig. So a previous study on the alcohol addiction has been in the animal model has been studied in rodents and also in pigs and in monkeys. And all of those studies show that, for example, in the rodents, they find that 20% of the rodent, they prefer alcohol over glucose, although they tend to drink more. And for the monkey, they also like to drink. But for our pig, we don't know whether they really like to drink. So what we did is that in the first stage, we do the induction. So we first pair the vodka, the real vodka, so with the apple juice. And then we gradually reduce the amount of apple juice. And then and at the end of the 3 months, and the animal, if we give the animal only vodka, and then they can drink at least 1 gram per kilogram per day. So when people, so here I'm showing you a very early video clip that, in here, the plate is containing the alcohol solution, and the pig already finished drinking the alcohol in here. But what you can see is that animal try to get up to reach the plate here and then to drink more. So the animal is clearly drunk. And he tried to reach the plate. Okay, so that's the first stage. So we know that some of our pig, actually most of our pig actually like to drink alcohol. So in the next stage, what we did is that I call it the enhancement and the shaping. So here we increase the amount of alcohol gradually, so the animal can drink from 1 gram per kilogram per day to 3 grams per kilogram per day. So this is actually a level of the binge drinking level in humans. So based on the standard is actually based on the BAC, the black alcohol concentration. So it's about 100 microgram at this level. So what you can see is that for our pig, they clearly can reach this level at the BAC curve. And then after that, we start the operant conditioning training. We train the animal to use the nose to push the bottom in order to get alcohol solution. And then at this stage, we increase the familiarity of the procedure. So on the third stage, we use the fixed ratio and also progressive fixed ratio schedule to detect their motivation of consuming the alcohol. So here is a short video clip that the animal is well trained. So here you can see that there's a red button in front of them, and there's a part underneath. So once he pressed the button for a certain amount of number, then he will get a 5 cc reward. So what you can see, animal is keep pushing the button. Okay, so because of time limitation, so let me go on. So here I'm going to show you some preliminary results. One is that we show so here is I show you the progressive fixed ratio at the x-axis, and the y is the number of pressing. So for the 10, it means that they have to press 10 times in order to get a 5 cc reward. And then after they get a 30 reward, we move on to the 20, and then we move on to the 30. So and then the different color represent the same animal at a different section. So what you can see is that in some of the section, the animal can press as high as 700 times in this. And in most of section, they can reach the 50, the fixed ratio 50. So here I'm showing you that the maximum ratio can reach of different animals. So you can see that the level they can reach is relatively stable. So and then in the future, what we are going to do is that we are going to use the radiosurgery to affect the region of the acute nucleus accumbens, and that's the image that's showing that we use the spatial MRI sequence called the FAGTIR. We can clearly identify where the nucleus accumbens is. So here a summary and the future direction. So the main summary is really from this figure is that for the focally delivered ion radiation of the low dose could induce durable and elevating metabolic activity without lesion in the brain tissue in the process. So the future direction is that now we only have one animal at each dose. So we want to larger sample size, and also we want to compare the gray and the white matter, and also the effect of SRS on neural activity like previous speaker. And also we need to establish the good animal model of different disorder, and also compare the technique with other neuromodulation tool. So as I say in the beginning, this project is funded by ZAP, and here are the team from National Taiwan University and the National Taiwan University Hospital. So some of the neurosurgeons are involved, and Fu Shaw is the PI, and for the PET team is Mei Fang Chen, she is the PI, and also we need a veterinarian team, and Yilin Chen is currently our veterinarian in this project. And also the animal team is Professor Zhu, that he is the one that involved the Li Song Minipig development. And also Kexin is my postdoc, and most of the MRI data analysis and PET data analysis is done by her. And thank you for listening. Thank you very much.
Time for a couple questions. You want to use the microphone.
Who's up for a question? Professor Halpern.
That was a really nice talk. It must be a challenge to develop this model. Strong work on doing that. I've only done it in a mouse, which is obviously a lot easier.
Yes.
In any case, I just was curious. In mice, alcohol is actually fairly aversive at first.
Yes.
And so it took us some time to provoke binge eating, or binge drinking, I should say, in our drinking in the dark model. Is that why you pair the alcohol with juice?
Yes, they clearly like the apple juice, include the taste and also the smell. So we didn't use other juice. We just picked the apple juice in the beginning, and it seems to work.
And they're just
so
mixed together, or is it two separate spouts?
we mix together. No, we
I
mix
see.
it together. So the animal is under the normal diet procedure. They are not water restrict at all, and also food restrict. They are just in a normal condition. In addition, we just give them the alcohol with the apple juice in the beginning, and then we gradually reduce the percentage of the apple juice until the percentage reduced to 0.
Understood. Thank you.
Congrats for your work. Very nice research. And I just wanted to wonder some observation on the MRI imaging. And I know you are a, N of 1 in each group dose. But I was kind of surprised that you didn't see any changes on T2, on doses ranging from 20 to up all the way to 60, because, it doesn't reflect lesioning necessarily. So it would be okay to have in healthy histology, but you didn't see in one animal breakdown of blood-brain barrier or any other changes that would structurally suggest the metabolic changes that you observed on PET. Also, if you could elaborate a little bit more on the bilateral changes, because you had M1 on the right as well, which makes sense when we think about network results. And actually, it's very enthusiastic that you can trigger that kind of reaction contralaterally if we consider, innumerable types of conditions that we may treat with unilateral radiation. So,
okay. So for the second question, so we know there's a lot of inter-hemisphere connection between the M1 as well, but what I found is that actually the lesion or the change in the MRI image is really local. So here I'm analyzed the region of the within a 7.5 millimeter of kilometer. So and outside of this region, let me show you one of the slides that I didn't show here. Okay, so on the left side is actually the T2 weighted image. So you can see that, and then on the x-axis is the slice along the color roster axis. It means that from the anterior to the posterior. And then in the middle is the range of the irradiation is about 7.5 millimeter. And what you can see is that when we move more anterior or more posterior, actually we didn't see any change in the MRI signal. So what I would say, so this result seems to suggest that the radiation effect is very, very local, and they seem to does not affect the region outside of the irradiation. And also, we didn't see any change really in the contralateral hemisphere. So that's also very interesting. But again, as I said earlier, we don't know whether the MRI technique is sensitive enough to pick up the change in brain tissue. So I think more study need to be done in the future.
Can I make a question? Is it okay? The response with 40 gray, apparently this is well described in the literature that it's almost a threshold of those that we start to cause damage. Okay.
Yes.
Happens this is it's not a point, it's a continuum that happens. I think Dr. Regis was very clear to say to you that it's a long time that the radiation would work. In our data that we did epilepsy, when we did 40 gray, we had this activation you described, but 3 months later, you had completely abolishing the seizure focus. Can you explain that?
Okay. So
A hard one.
let's I only can speculate the outcome that you just mentioned, but I'm not really sure what's really going on, especially with only one animal in the 40 gray here. But it seems there's a border here that it's between the 40 and 60, the gray area. It seems that the PET signal doesn't change much, and the level is very similar to the baseline, although you see a little bit elevation at the 40. And in terms of histology, we did see some small injury in the 40s. So and one of the possibilities is that the animal that received a 40 gray could have the injury in the tissue earlier, or maybe 6 months after the radiosurgery. As I said earlier, this is the histology we done about 1.5 years after the radiosurgery. So maybe the brain has the ability to repair itself. So we see the damage is smaller in the 40 gray. That's one my speculation.
Yeah, thank you so much for this inspiring talk. I just have one question. You saw, or you radiated the primary motor cortex, so and your increase in metabolic rate could also suggest that there's an increase in neuronal activity. But as it's affecting M1, can you maybe look at the pig's behavior? Was there kind of higher impulsivity, or were they just moving around more? Because I think that might at least behaviorally close the link between higher metabolic rate and actually higher activity of M1.
Okay, so I did mention this earlier, but I mentioned the condition in the higher dose. But for the lower dose, because it's just an observation, we didn't really measure for example, do the step analysis or something like that. So I really cannot tell is any change in terms of their movement at this stage. Yeah.
I think we need to move. Okay?
Okay. We'll hope,
you want to say?
Chun-I, we'll hope you'll join us for the roundtable and for further discussion.
Okay. It will be 4 a.m. for me. I'll try to join.
It's for a good cause, Chun-I.
Thank you.
Oh,
So
Sorry
I
about
should
that.
stop sharing my screen. Hold on.
got a quick nap. But anyway, if you could join us, great. If not, you gave a brilliant presentation. Thank you, and I'm sure thought-provoking. A lot of people in the room are going to be thinking about it over the rest of this afternoon. So thank you, Chun-I, and with luck, we'll see you later.
John. Thank you, Brad. Thank you.